Thank you all for inviting me to talk to you today. I'm going to talk about computational biology at the exascale. I'll say a little bit about cloud computing as well. And I'm going to talk about this partly because I think these are a particularly challenging class of algorithms that come up in this application domain that are quite different than a lot of other scientific problems. So specifically, I'm really going to be looking at genomic analysis and what is called metagenome analysis, and I'll say a little bit more about this in a minute. This is a big data problem in the sense that the amount of genomic data is, of course, growing exponentially. This is looking at the drop in the cost of sequencing. We're going to need very scalable algorithms, and I'll talk a lot about that, and also big machines in order to analyze some of these data sets. So how big are these data sets, and why do you need even an HPC system? Well, certainly the tradition in a lot of metagenomes is looking at, in a lot of genomics is done on shared memory machines, and that limits the size of the data set you can analyze. And what I'm looking at here is microbial data. And that comes up, you heard about the importance of the human microbiome, your gut microbiome, and the impacts on health, but also in the environment is very important for understanding various aspects of environmental impacts and things like that, agricultural and so on. And there are a number of questions that come up, such as what do these microbes do? This data set is also a very large and growing data set from the microbial data. And what are some of the kinds of scientific questions that people ask for these, and how big are the data sets? Well, this is a data set that people that use the Joint Genome Institute at Lawrence Berkeley Lab and the supercomputers at NERSC using our tools that I'll be talking about in terms of the algorithms have analyzed. The question is what happens to microbes after a wildfire? This is a 1.5 terabyte data set. Questions like what do these microbes do? And in this case, the scientists are using it with isotope tracing data, and that's an 8 terabyte data set. And finally, a study that's going on now, which is looking at how do carbon and metabolism in freshwater lakes change across 17 years, and that is a 25 or 26 terabyte data set. So there are some very large data sets, and these are very complex genomic analysis problems to solve, because when you scoop up some of this water, it might have thousands of different species in it. Each one of them, when you send it through the sequencer, their genomes get fragmented into little pieces. And we're looking at short sequences for a lot of the work that we're doing. So say 150 characters, and you're trying to turn that into a complete or nearly complete genome by basically fitting the puzzle pieces together, finding overlapping pieces of those strings, and then finding out how they match. Now, the other thing that's a challenge here is looking at the exascale architectures. I don't need to tell you all about the end of Dennard scaling and the pending end of Moore's law, except to say that when we started talking about exascale architectures, we had thought that this project would probably be focused on things like many core architecture, such as the Knights Landing line of processors. And instead, all of the DOE systems, and this shows the 3 systems planned for the Office of Science, are based on GPUs. And they're all HPE systems, and they're based on either NVIDIA, AMD, or Intel GPUs. So this means that we need to now map these problems onto GPUs and onto, of course, a large amount of data parallelism. And so the 2 sort of themes that are going to come up in this talk in terms are, well, or maybe 3, is the irregular nature of the algorithms, the need to use fine-grained and asynchronous communication, and really to optimize communication in various ways to avoid doing communication, and finally, the need for data parallelism to take advantage of the GPUs. So a little bit about the algorithms and software. I'm not going to try to describe these complete applications and what they look like. I'll just give you a sense of what some of the computational kernels are, or the motifs, the patterns that come up in genomic analysis. And this is a list and a paper that we put together on these motifs that identifies really 7 patterns that come up in genomic analysis. This is very close to the set of what are called the 7 giants of large-scale data analysis, of big data analysis from a National Academies report. And but here you'll see some examples from problems such as assembling the genomes, that is taking the fragments and putting them together into longer pieces, computing the distances between different pieces of a genome, the genes, the proteins, the other sort of the k-mers or k-mer profiles, and clustering things and annotating things and so on. So the first technique that is very important in a lot of these applications and comes up over and over again in different application scenarios is hashing. And this is used, for example, to count the frequency of various substrings. And we do this for a number of different reasons. One is to get sort of a profile, an approximate sort of signature of what a particular metagenome might look like by looking at what its k-mer profile looks like. We also use it to filter out reads. So if there's a k-mer, a fixed length string, so a k-mer is a fixed length string that appears only once in the data set, then we might determine that that's likely to be an error in the that was produced by the sequencer. So when we're looking for these k-mers, what we'll do is we'll take the input strings and assume that we've read over the genome multiple times, so we should have multiple copies of any particular location. We'll pull out these fixed length strings and then hash them and use that to count them. And we'll build a data structure, a hash table with them, which we can certainly use for counting. We also use more compact data structures. We do use things like Bloom filters in order to reduce the total amount of storage, because there are a lot of singletons in here that we don't care about, at least for a lot of the analyses that we're going to do. But then we also will store with each key, with each k-mer, that's the hash key, the left and right character that appeared in the original input string with that, so that we can then go back and glue them together into these longer sequences. So we filtered out the errors, and then we can reconstruct the genome, hopefully without too many branches from those k-mers with their left and right extensions. So this builds a large distributed hash table. If you think about the, say, 8 terabytes of data or the 25 terabytes of data, if you have a typical k-mer length is going to be, say, between 50 or 21 and 100, that we're going to blow up our data set size by another factor of 50 to 100. We won't do that kind of all at once. We'll try to never store that in memory, but that gives you a sense of how large these kind of intermediate data structures are going to be when we compute these k-mers. Once we compute, we can save, once we've hashed them, we save only one copy of each unique instance of a k-mer. That will be a much more manageable data set. That's about the size of the output of the data, not the size of the input data set, or certainly not as large as the k-mer expanded version. So we have looked at both bulk synchronous versions using MPI, where you read a bunch of data, you compute you parse the k-mers, you exchange them, you build a local hash table and or a partition of the hash table, and also in an asynchronous algorithm that's based on UPC++, a partition global address space language, in which you do one-sided communication, and you do things like RPCs, which are used extensively in the code to sort of remotely insert a bunch of elements into a hash table or a local piece of the hash table. And just looking at the difference in performance between the bulk synchronous version, this is the blue line here, and the red line. So the blue line has a Bloom filter, which is really used to reduce space, not to reduce running time. And that compares then with the red line, which is, with the yellow line, the red line, which is also the UPC++ version with the Bloom filter as well. So those two both have the Bloom filter, and it's actually faster without, but we often use it with the Bloom filter turned on in order to avoid building such a large intermediate data structure. So you can see the advantages of using these asynchronous communication. And the way I like to think about it is a phrase that actually came up decades ago when people were programming the SIMD machines, which is to keep all the wires busy all the time. And that's really what you're doing here, is you've got this very irregular all-to-all sort of pattern, but you don't want to just put it into bulk synchronous phases, because then you're wasting your wires, which are the most expensive kind of thing that you're trying to optimize for. Now, can you do this kind of hashing and counting on GPUs? You certainly can. It is not the most natural to think of in a data parallel style, but you can take a chunk of them and put them into the hash table. This graph on the left shows the CPU implementation running on 64 nodes that was dominated by the time to count the k-mers, the local work, and counting and parsing the yellow and the blue. And this little red piece was the communication piece. So what's a GPU? As some people would say, a GPU is something that takes a compute-bound problem and turns it into a communication-bound problem. This is the same red sliver here now expanded in the GPU-optimized version, where the compute time has basically disappeared and all you're left with is the communication time. This is a bulk synchronous version. There's now an asynchronous version of it as well to try to better overlap the communication and computation. The other thing, though, that's done besides overlapping is to try to see if we can reduce the total volume of communication. And here, there are some application level tricks that involve looking at things like hash functions that are going to give you better locality and therefore put a chunk of k-mers that are nearby each other in both the final answer, but also in the input strings nearby each other. And then rather than shipping each separate k-mer, you ship a contiguous string that gets fully parsed on the remote side. And you see some speedups from that on the right-hand side as well. Now, the second data structure that comes up a lot, motif that comes up that I'll talk about now, is graphs, which are also quite different than what you might have seen in, say, scientific simulations. And the graph problem I'll talk about here, although there are several of them that come up across the different microbial analysis projects. This one is what we call contig generation. And that is, once we've put these k-mers into a hash table with their left and right extensions, you want to go and build the connected component as a string that reflects what is that contiguous sequence of things. And that takes the k-mers and then turns it into these contigs. And that's, you can think of it as a depth first search that starts at every possible position within the graph, every vertex, but hopefully non-redundantly. So you don't want to do a search on something that's already been searched from another point. And that's really what happens. And it's done using UPC++ again for this sort of this asynchronous fine-grained communication. And there's a certain amount of caching optimizations that we can use for certain cases, and also using GPUs here. And this shows the benefit of the GPUs, that this is the CPU running time in blue and the GPU running time in red on a number of different nodes. So again, a problem that doesn't seem like it's necessarily a very good match for GPU, but turns out to actually speed up quite effectively. The last thing I'll really talk about in detail here is genome alignment or string alignment. And this comes up a lot in both, in certainly in genomics, but in some other data analysis problems where you're looking for inexact matches between different strings. So you may have heard of the Smith-Waterman algorithm, a dynamic programming algorithm, where you've got two strings here across the left and the top, and you're trying to figure out what is the best path through it that minimizes the number of insertions and deletions to make one string equal to the other string. So this diagonal part of the path, the GAT, are the matching things. So those are kind of free steps through the alignment. And then we have more expensive insertions that look like a horizontal, or deletions, a vertical line. And so we, in the worst case, can search this entire matrix in order to find the optimal path through these two strings that will give you the minimum edit distance between them. And in practice, we often, because these strings are going to be much longer, we will cut them off so that we don't actually search near the outside, because we don't care about really poor matches between two strings. Those are just not going to be considered an alignment. But the other thing that comes up in practice is that we want to do many of these alignments, and that's fortunate, because even though the strings get longer, even at 150 characters, or that you often do not want, you don't want to pay for that order n squared computation. And so what you can do is you can batch a number of them together and then run those on the GPU. And here the red, yellow, and orange bars are looking at GPUs, and 2, 4, and 8 GPUs respectively. And the two different blue lines are two different CPU implementations that take advantage of some of the heuristics to cut off the search because of the cost. But what you can see here is that this is in giga updates per second. So that's the number of updates of that matrix. And so in terms of giga updates per second, so up is good here. The GPUs, you can see, have a significant advantage in this alignment problem, where it's a batch alignment problem. And the last one of these, I guess I will talk about one more in just a little bit of detail, and that is looking at this problem of that is sometimes called generalized n-body. So this is some kind of an all-to-all computation where, for example, in traditional n-body calculations, you're looking for all, in the most naive setting, you're looking for forces between all pairs of particles, for example. And here what we're going to do is look for alignments between all pairs of strings. So it has the same sort of a feel. It can be done on a single data set aligned to itself, or in general, it can be aligning some set B with some other set A. Now, as with an n-body calculation in physical simulations, you don't want to actually compute all n squared alignments here. So this is a different n squared between all pairs of things than the alignment algorithm itself. But it's still, given that alignment is expensive, you want to only run alignment when you think there's a pretty good chance of a match. And so one of the ways in which that is done is by filtering out and only running alignment if there is a common k-mer that is contained between these strings. We do that once again by building a hash table with, say, the strings, all the k-mers from the strings in A, and then look them up from all the k-mers in the strings of B and look for things that match. And this can also be done in a bulk synchronous style, which has both a larger running time, a somewhat larger running time, although they're comparable, and a larger memory footprint than an asynchronous implementation that is just going and sort of grabbing the things that you need at a given point in time, and sometimes a set of things. So this shows both the running time and the performance advantage, and the memory footprint advantage of the asynchronous version. Now, we also see a lot of graphs inside of these algorithms that are actually going to be represented as sparse matrices. So just to continue with the previous example, if we've got a sparse matrix that stores k-mers as the key, so this is the k1 through k5 here on the bottom, and you see, and you're storing a read, which is the list of all reads that contain that k-mer. And so we can think of that as now as a sparse matrix, where in the read dimension, we kind of blow it up and there's a non-zero whenever that read contains that given k-mer. We can multiply that matrix times its transpose and get out a sparse matrix that is a read by read sparse matrix. And that tells us which pairs of reads contain a common k-mer and should therefore be aligned. And we can run these matrix-based algorithms now in more of a two-dimensional decomposition. And this shows a version of this aligner called dBella, both the original 1D version that was both the synchronous and asynchronous version, and now the 2D version that uses this more matrix perspective and does a two-dimensional decomposition of those matrices. Now, the last thing I'll just comment on here briefly, and I talked more about this in the panel session, is what about clouds, and can we run these kinds of algorithms on clouds? And I would say that the conclusion about 5 years ago when we started the this project to do genome assembly, the Exabiome project, was absolutely not. We were looking at cloud type of platforms, and this is looking at something with an Ethernet switch, both a 1 gig and a 10 gig Ethernet switch. And what you're seeing here is the slowdown going from one node to two nodes. And we sometimes do see a little bit of a slowdown, but not this dramatic, and it would take you a large number of nodes in order to try to ever recover that 18x slowdown that you see here. So you need a much higher speed network than you got on the cloud systems a few years ago. There are now actually pretty commonly HPC instances in the commercial clouds. And so we're seeing much better communication performance. This is a micro benchmark looking at all-to-all latency just on two nodes, but nevertheless, what you can see is that this AWS system is actually very competitive with Cori. In fact, there's, I think, some places where it's actually faster depending on exactly which version you're using. So this also, because of the fact these HPC instances allow you to do co-scheduling of the jobs, and you really get dedicated instances. They're very expensive, but they are give you comparable HPC performance for at least for very small scale problems. And we're studying right now how large we can scale these to, and also looking at a number of different application benchmarks on different size and different types of cloud instances. So to wrap up, communication avoidance is still a very important problem in these irregular computations, which are represented by these genomic calculations. Traditionally, when people, because of the complexity of these algorithms and the fact that they're building things like shared hash tables, they were often run on large shared memory systems. And I think we've been really pushing the envelope into these distributed memory systems that are both the terabyte shared memory systems are both too small and too slow. These problems map well onto distributed memory hardware if you've got very lightweight communication, and even to GPUs if you've got, by thinking about the algorithms in a way of batching these very small lightweight tasks onto the GPU to give you these data parallel-like operations. Communication avoidance is still key at their application level techniques that I tried to give you a couple of examples of in terms of in the K-mer mapping, trying to get hash functions that are going to give you better locality. For example, bandwidth reduction. So judicious use of caching in some of the algorithms to avoid multiple times looking up the same remote value. For example, when you're aligning something against something else, and but also using these sort of 2D or higher dimensional algorithms by dividing up the iteration space of the computation rather than just dividing up the data structure and spreading that out over the processors. And finally, hiding latency as opposed to packing things together to reduce the cost of latency. Those two will trade off against each other. And I think our approach is to try to basically keep all the wires busy all the time, which means doing a better job of hiding latency and using very lightweight communication to keep those wires busy. So with that, I will wrap up and thank you very much for your attention, and I'm happy to take questions.